All right, let's dive right in. Today, we're taking a guided tour into the world of some of Earth's oldest and honestly most successful life forms, bacteria. These tiny single-celled organisms are literally everywhere. They are true masters of survival and just microscopic marvels of engineering. So, let's get our microscopic viewfinders ready and jump in.
Okay, first things first, let's get our bearings. Bacteria are prokaryotes, and the biggest difference between them and our own cells, eukaryotic cells, is right here. See, our cells have a proper command center, a true nucleus, all nicely wrapped in a membrane. Bacteria? Nope. They keep their genetic blueprint just kind of free-floating inside. And then there's the size. We're talking a whole different scale. It's like comparing a tiny, super-efficient studio apartment to a sprawling mansion. So here's our roadmap for this journey. We'll start with the basic shapes, the blueprint. Then we'll check out their external toolkit for surviving out there. After that, we're breaching the Great Wall of the cell, taking a peek inside the factory floor. And finally, we'll see what the day-to-day life of a bacterium is really like.
So, let's start with the absolute fundamentals. Before we get into all the complex machinery, what do these microscopic architects actually look like? Because, as you'll see, their form is totally connected to their function.
For all their complexity, bacteria generally come in three main flavors. You've got the little spherical ones, the cocci, the rod-shaped ones called bacilli, and the twisty spiral forms. But it's not just about their individual shape. Look at the cocci. They can form these long chains, like streptococcus, or they can clump together in these grape-like clusters, like staphylococcus. These arrangements are super important. They decide how bacteria build colonies, talk to each other, and even how they make us sick.
Okay, so let's move to the exterior. A bacterium is always out there, interacting with its environment, and it's got this incredible set of tools on its surface to help it move around, stick to things, and protect itself.
Meet the bacterial outboard motor, the flagellum. This whip-like tail is an absolute marvel of nanoengineering. It's built from a protein called flagellin, and it's anchored into the cell by this complex little motor, the basal body, that lets it spin around like a propeller, pushing the bacterium through whatever liquid it's in.
So if flagella are for moving, pili are for sticking. Think of these shorter, hair-like bits as tiny grappling hooks. They let bacteria latch onto surfaces, like, say, the cells inside our bodies. And here's where it gets really clever. Some bacteria have these special sex pili. They can actually form a little bridge to another bacterium and transfer small packets of DNA, sharing survival tricks like antibiotic resistance. Pretty wild, right? And the final piece of gear on the outside is the capsule. You can think of this slimy outer layer as an invisibility cloak. It's made of sugars, and it helps the bacterium hide from our immune cells so they don't get eaten. It also keeps the cell from drying out. So it's basically a shield and a raincoat all in one.
All right, we've covered the exterior. Now it's time to peel back those layers and look at what is, arguably, the most defining feature of most bacteria, the cell wall. And this isn't just a simple barrier, it's a complex, multifunctional suit of armor. Now this right here shows the great divide in the bacterial kingdom. There are two main architectural styles for that cell wall. On one side, you have gram-positive bacteria. They've got a really thick, tough wall of something called peptidoglycan. Think of it like thick brickwork. On the other side, you have gram-negative bacteria. Their peptidoglycan layer is much thinner, but they add a whole other outer membrane on top of it, which has some really unique stuff in it.
And this is that unique stuff I was talking about, lipopolysaccharide, or LPS. You only find it in the outer membrane of gram-negative bacteria. Now, it's great for the bacterium structure, but for us, it's also known as an endotoxin. That lipid A part specifically can set off a massive inflammatory response in our bodies if it gets into the bloodstream. It's a huge reason why gram-negative infections can be so dangerous.
So how do we tell these two different wall types apart in the lab? Well, we use a technique that's over a century old, called the Gram stain, and it brilliantly exploits these structural differences. First, you douse all the cells with a purple dye, crystal violet. Then you add iodine to make the color stick. Now here's the key step, the alcohol wash. In gram-positive cells, that super thick wall traps the purple dye. But in gram-negative cells, the alcohol just dissolves that outer membrane, and the thin wall can't hold on to the dye, so it all washes out. To finish, you add a pink counterstain, which colors the now clear gram-negative cells. So purple for positive, pink for negative. It's all because of the wall. And if you ever doubted how vital that wall is, just look at what happens when you get rid of it with something like penicillin. A gram-positive bacterium turns into this fragile little ball called a protoplast. A gram-negative one becomes a spheroplast. In either case, without that rigid support system, the cell is just incredibly vulnerable and can pop like a water balloon.
We've breached the wall. Let's head inside to the cytoplasm and see how this super efficient, minimalist factory runs, especially since it doesn't have all the fancy departments and cubicles you'd find in our own cells.
Which, of course, brings us to a really important question. If there's no official membrane-bound office for the CEO, no nucleus, where does a bacterium store its most precious asset, its DNA blueprint?
And the answer is the nucleoid. Now, this isn't a true organelle. It's more like a designated area, a dense, tangled region in the cytoplasm where the bacterium's single circular chromosome is all packed together. It's the command center, just without the walls of a corner office. All the information needed to run the entire factory is right there.
And what's just as amazing as what's inside a bacterium is what's not inside. Think about it. It gets by without any mitochondria for power plants, no endoplasmic reticulum for manufacturing, no Golgi apparatus for shipping and packaging. It's a true masterclass in minimalism. All those complex jobs just happen right there in the cytoplasm or at the cell membrane. It's the ultimate open plan office.
So for our final section, let's zoom out a bit. We've seen the individual machine, but now let's look at the whole operation. How these cells live, how they grow as a population, and how they show off their incredible resilience.
You see, when bacteria find a new home with plenty of food, they don't just immediately go nuts. They follow this really predictable growth curve. First, there's the lag phase, where they're just getting adjusted, kind of tooling up for what's next. Then comes the log phase, and the population just explodes. 1 becomes 2, 2 becomes 4, and on and on. Eventually, though, as food runs low and waste builds up, they hit the stationary phase, where growth and death rates balance out. And finally, as things get worse, they enter the decline phase.
You know, part of their incredible adaptability is in how they breathe. Some, called obligate aerobes, need oxygen to live, just like us. Others, the facultative anaerobes, are super flexible. They'll use oxygen if it's around, but if not, no problem. They can switch over to fermentation. And then you have specialists, like microaerophiles, that like just a little bit of oxygen, not too much. This amazing diversity is why they can thrive pretty much everywhere on this planet.
But what happens when things get really bad? I'm talking no food, extreme heat, radiation? Well, some bacteria have an unbelievable ace up their sleeve, the endospore. Now, this isn't about making more bacteria. It's a pure survival move. The cell packs its DNA and a few essential bits into a super tough, dormant core. It's like an emergency panic room that can withstand almost anything, just waiting, sometimes for centuries for conditions to get better before it comes back to life.
And that brings our tour to a close. We've seen a blueprint for mind-boggling efficiency, a toolkit for survival, and strategies for enduring the absolute unimaginable. These tiny machines have been on our planet for billions of years, adapting and thriving. So it kind of leaves us with one last thought to chew on. What can their simple yet profoundly powerful survival strategies teach us about resilience in our own, much more complex world?